Because IFE2 is the most abundant EIF4E isoform in somatic C. elegans tissues, these findings suggest that reduction of protein synthesis, specifically in the soma, extends lifespan. Depletion of other somatic or germline-expressed EIF4E isoforms does not cause similarly pronounced effects on nematode lifespan. However, depletion of IFE1 during adulthood results in modest adult lifespan extension, suggesting that IFE1 also modulates longevity. Post-developmental elimination of other translation initiation factors or their regulators has analogous effects on the longevity of the nematode, reducing the levels of the scaffold protein EIF4G or the EIF2 beta subunit by RNA interference, RNAi, during adulthood leads to a nearly 30% increase in lifespan. Similarly, using RNAi to reduce the levels of several ribosomal proteins or the ribosomal protein S6 kinase, S6K, specifically during adulthood, extends nematode lifespan. In all these cases, the rate of protein synthesis in RNAi-treated animals was reduced in comparison with that in wild-type controls. In addition, many genes encoding components of the EIF complex and components of the 40S and 60S subunits of the ribosome were recovered in an RNAi screen for essential genes that extend lifespan when inactivated post-developmentally. One is the origin of lifespan extension by reduction of protein synthesis. IFE2 depleted mutants are considerably more resistant than wild-type animals to cellular oxidative stress induced by the herbicide paraquat, a methyl vialoge, or the inhibitor of the respiratory chain, NANAN3. Furthermore, IFE2 deficiency increases oxidative stress resistance and extends the lifespan of MEV-1 mutants, which experience chronic oxidative stress due to their lack of the cytochrome B large subunit in complex 2 of the mitochondrial electron transport chain. Thus, depletion of a specific EIF4E isoform, IFE2, expressed in somatic cells increases oxidative stress resistance and extends lifespan. The fact that animals with compromised protein synthesis have increased resistance to several stressors further supports the link between translational regulation and general stress responses. Such animals are more resistant than wild type to various stresses, such as heat shock, oxidative stress, UV irradiation, or starvation. Interestingly, EIF4G levels are reduced during the Dauer stage, an alternative nematode larval form that is normally induced by stress conditions such as crowding and food deprivation. Dauer larvae do not feed, and they have slowed metabolic rates and live longer than reproductive adults. Reduced protein synthesis might contribute to the longevity of these animals. If the turnover and replacement of damaged proteins is required to avoid senescent decline, how does lower protein synthesis augment stress resistance and extend lifespan? mRNA translation is one of the most energy-consuming cellular processes, devouring an estimated 50% of the total cellular energy, depending on the organism and cell growth state. For example, mRNA and ribosome biosynthesis, two processes controlled by insulin IGF-1 and TOR signaling, are highly energy-consuming. Under favorable conditions, yeast cells synthesize 2,000 ribosomes per minute to maintain robust growth. Therefore, reduction of protein synthesis rates under unfavorable stress conditions would result in notable energy savings. Indeed, global translation is reduced in response to most, if not all, types of cellular stress. This energy could then be diverted to cellular repair and maintenance processes, thus contributing to longevity. However, a decline in turnover rates would delay the removal and replacement of damaged proteins, thus contributing to senescence. Indeed, protein synthesis is an essential process that has an impact on all cellular functions. Complete elimination of key mRNA translation factors cannot be tolerated. Genes encoding these proteins are essential for growth and development and are usually highly conserved in evolution. To investigate these questions without incurring the deleterious consequences of interfering with the expression of essential mRNA translation factors, researchers have used RNAi to target the corresponding genes after completion of development and during adulthood in C. elegans. These experiments reveal that reduced expression of genes that are essential early in life actually enhances longevity when RNAi is initiated later into adulthood. Late-onset interference usually also results in increased stress resistance and decreased fecundity, indicating a possible trade-off between somatic maintenance and reproduction. Such a trade-off is postulated by the antagonistic pleiotropy theory of aging. This theory proposes that pro-aging alleles with adverse effects late in life, after the reproductive period, are nevertheless maintained in the population by natural selection owing to their beneficial functions early in life. The totality of these observations suggests that perturbing protein synthesis is a double-edged sword, promoting either longevity or senescence, depending on whether or not a certain threshold is exceeded. A delicate balance exists between the maximal longevity benefit and the detrimental impairment of protein metabolism. Several cellular signaling mechanisms converge to influence the rate of mRNA translation in response to a variety of stimuli by modulating the activity or the availability of important translational regulators. The insulin IGF-1 pathway modulates aging in a variety of organisms. For example, downregulation of insulin IGF-1 signaling by mutations extends lifespan in C. elegans. This extension depends on the activity of DAF16, a forkhead FOXO transcription factor. DAF16 controls a wide variety of downstream targets, thereby affecting stress resistance, fat accumulation, fertility, and metabolism. In Drosophila, mutations in both the insulin-like receptor, INR, and the insulin receptor substrate, Chico, prolong lifespan in homozygous female flies, with evidence that D-FOXO is also required for longevity. In the yeast S. cerevisiae, mutations in the protein kinase SCH9, suppressor of CDC25, which is a functional ortholog of S6K and also shows sequence similarity to ACTI, also known as protein kinase B, PKB, increase lifespan and stress resistance. CH9, together with the TOR kinase, mediates the effects of caloric restriction on aging in yeast. In mammals, separate receptors for insulin and IGF-1 mediate distinct signaling events in different tissues. Insulin coordinates cellular metabolism, whereas IGF-1 regulates growth and differentiation. Mutations in either receptor gene or in upstream genes that regulate insulin or IGF-1 levels extend lifespan. For example, the Ames and Snell dwarf mice, which have low levels of serum insulin, IGF-1, and growth hormone, live significantly longer than their normal siblings, as do dwarf mice mutant for the growth hormone receptor. The underlying molecular mechanisms that mediate these effects on lifespan are not clear. However, the rate of protein synthesis is decreased significantly in long-lived Snell dwarf mice. Downregulation of mRNA translation is the result of reduced insulin IGF-1 signaling through AKT, PKB, and P38 MAPK, which in turn control key translation regulators such as the mammalian target of rapamycin, mTOR kinase, ribosomal S6K, BEIF4E kinase, MNK1 mitogen-activated protein kinase interacting kinase, and the translation initiation factors EIF4E and 4E-BP1. A similar mechanism mediating protein synthesis reduction appears to operate in long-lived Ames dwarf mice, in which PI3-ACT mTOR signaling is attenuated. Therefore, low protein synthesis is a common denominator of both Ames and Snell long-lived dwarf mice. TOR, an evolutionarily conserved serine-threonine kinase, has emerged as a central regulator of cell growth and proliferation, development, metabolism, and aging. Genetic and biochemical studies in yeast, C. elegans, Drosophila, and mammalian cells have identified several upstream and downstream components of the TOR signaling pathway. TOR activity is regulated by four major input signals: nutrient and energy availability, growth factors, and stress. Low insulin IGF-1 signaling, nutrient or energy deprivation, and stress converge to downregulate the activity of TOR. In C. elegans, TOR deficiency causes developmental arrest, but these arrested animals live longer than wild-type arrested worms. Similarly, knockdown of TOR by RNAi in adult animals increases their lifespan compared with that of wild-type. Genetic data suggests that the TOR pathway can also mediate the life-extending effects of dietary restriction. Therefore, the TOR and insulin IGF-1-like signaling pathways might integrate nutrient sensing and nutrient uptake to influence adult nematode lifespan. How might TOR influence lifespan? TOR interfaces with several cellular processes such as DNA transcription, mRNA translation, protein turnover, autophagy, and actin cytoskeletal organization, among others. However, one of the best characterized functions of mTOR is its ability to regulate protein synthesis. Biochemical analysis has revealed an orchestrated sequence of events that promotes efficient protein synthesis under conditions that activate mTOR. Two key downstream targets of mTOR are S6K and 4E-BP1. Deletion of either dTOR or DS6K in Drosophila results in small cell size and severely reduced body size, similar to the effects of loss-of-function mutations of positive regulators of the insulin IGF-1 pathway. S6K phosphorylates the 40S ribosomal protein S6 and selectively promotes the translation of 5-TOP mRNAs, a specific subset of mRNAs containing a terminal oligopyrimidine tract. Interestingly, these mRNAs often encode ribosomal components and translation elongation factors. 4E-BP acts as a translational repressor by competing with EIF4G for an overlapping binding site on EIF4E. Phosphorylation of 4E-BP promotes its dissociation from EIF4E, enabling recruitment of EIF4G and EIF4A translational factors to the mRNA cap structure. mTOR also regulates other translation initiation factors, such as EIF4GI and EIF4B, in addition to elongation factors such as EEF2. In addition to downregulation of translation under nutrient deprivation or environmental stress, such as heat shock, osmotic shock, or UV irradiation, reduced TOR activity induces autophagy, a lysosomal catabolic pathway for the degradation and turnover of proteins and organelles. It is noteworthy that autophagy is elevated in long-lived DAF2 nematode mutants, and BEC1, the worm homologue of mammalian Beclin-1, a protein essential for autophagosome formation, is required for lifespan extension in these mutants. Thus, the TOR and insulin IGF-1-like signaling pathways might influence lifespan by relaying nutrient signals to modulate protein synthesis and turnover.